So friends, welcome to my YouTube channel Govind Narayan Purohit, wherein we are discussing the different topics of theriogenology. And in our today's talk, we discuss something about semen collection from male animals and its evaluation. Friends, I express my sincere thanks to all of you who have subscribed to my channel, and uh, if you have not already subscribed to my channel, please do subscribe to it so that you receive our videos very fast. So friends, we talk first about the semen collection. Semen has been collected from many species including cattle, buffalo, sheep, goat, stallions, boars, chicken and turkeys, and has been widely used for genetic improvement. In any species, there are two principal indications for collecting semen: as a prelude to artificial insemination, which often involves short or long term semen storage, and then second, to assess the potential fertility of a male and provide diagnostic information in cases of infertility. Now, there are different semen collection techniques. The most common technique is use of an artificial vagina. Then, by digital manipulation, semen can be collected from dogs, and by using electro-ejaculation, one can collect semen from rams, bulls, and a few of the other species. The technique used depends on the species being collected and the disposition of the individual male. Now, first we talk about the artificial vagina. Artificial vagina are used to collect semen from vagina are that the male be conscious, not significantly frightened of people, and more interested in ejaculating than in killing humans. And AV uses thermal and mechanical stimulation to stimulate ejaculation. An AV is made of an outer case which is of a hard plastic. Then there is a inner liner uh, which is placed inside the AV and folded on both the sides. Then there is a water jacket from through which the hot water is filled in the AV, which keeps the temperature of the AV warm. Then there is an inner liner. This is folded on one side, and on one side it is attached to the collection tube. So here this is an AV which is commonly used for collecting semen from bulls. This is the hard cone, and this is the wall. This is the these are the attachments: the inner liner, the cone, and then the hose for filling in water and air inside an AV. An AV is composed of a tube with an outer rubber lining that would hold water, into which is placed an inner liner that is lubricated just prior to use. The outer liner is filled and pressurized somewhat with water at 42 to 48 degrees Celsius. Now, to collect semen from bulls, the bull is brought near another male. A training is given to bulls during young age to donate semen by mounting on uh, estrus cows, or maybe it is better to herd mates, to make them mount on herd mates, or if they are trained to donate semen on an artificially prepared uh, male, then it is also better. To collect the semen, the artificial vagina is prepared, and the temperature, which is particularly prominent recommended for the species, is maintained by by filling it with hot water. To collect the semen, the male is allowed to mount, and the penis is directed into the AV where he ejaculates. You can see in these pictures, before mounting, one or two false mounts would increase the quality and quantity of the semen, and this is how a bull is made to mount. When he mounts and the penis is protruded, the penis is guided into the artificial vagina. It is best to allow the male to thrust into the AV rather than trying to slide the AV onto the male's penis. Then uh, the in artificial insemination, centers or where semen is routinely collected. Bulls are typically collected 2 or 3 times per week with 2 or 3 ejaculates per collection day. Now this is how the in the buffalo the artificial vagina used for cattle is commonly used, and the tris buffer is used for dilution of the semen. This is how the buffalo is mounting on another male buffalo, and the collector he collects the semen through an artificial vagina. Artificial vagina used for semen collection from different animals are shown here. From rabbits, a smaller one, then a director cone used for semen collection from dogs, and then this is the artificial complete assembly of the artificial vagina for semen collection from bulls. The because semen and the spermatozoa are liable to thermal stress, so this should be covered with a outer insulation jacket. In this is how the semen for semen collection from stallions, an artificial vagina is being prepared. This is the Missouri model. It is filled with water, and then stallion semen, it contains plenty of gel, so a filter is applied, and then this is how the AV is now ready. It a outer insulation jacket has been applied. Then Y-loop is used for lubricating the inner part of the artificial vagina. Many models, the Colorado, Missouri, Lane, Hanover, Nishikawa, and Cambridge, are available for collecting semen from stallions. In most models of AV, air is added by a valve in order to regulate the pressure, because in stallions the pressure seems more important. This picture shows again the semen collection from indigenous stallions. A Marwadi stallion is used for collecting semen at the National Research Center on Equines at Bikaner. Now, semen collection from male camels is done in a sitting position, and the we have talked about the peculiarities of the camel penis, and when it becomes straight, the operator he guides the semen into the artificial vagina, and the artificial vagina used for collecting semen from bulls is used for camels. Then smaller models of artificial vagina, as shown in this picture, are used for semen collection from bucks and also from rams. Then digital manipulation is used for semen collection from male dogs, and pressure and massage of the penis is used. The semen is collected through a director cone, especially in dogs, pigs, and turkeys. Massage of ampullae and seminal vesicles can be used for semen collection from bulls, but it will give more fluid volumes, and the sperm concentration would be low. Then electroejaculation involves applying a series of short low voltage pulses of current to the pelvic nerves, which are involved in the ejaculatory response. Semen can be collected from bulls and other species, but it should not be used in stallions. The advantage of electroejaculation is that semen can be collected from bulls with leg problems, but the semen volume is large and sperm concentration is low. So, in general, the artificial vagina is the method of choice for collecting semen. Now, then, semen handling. The sperm are motile and vigorous cells, but also fragile and susceptible to damage and killing by several environmental conditions. They are likely to be damaged by exposure to toxic chemicals, by thermal stress, and sperm are sensitive to both heat and cold. To avoid thermal stress, the collection tube end of the artificial vagina should be covered with an insulating cone. Additionally, microscope slides, cover slips, stains, extenders, and pipettes used to handle and examine sperm are best maintained on a warming plate prior to use, as shown in this picture. This is a warming plate where everything, the temperature of everything is maintained by placing the slides, etc., on this warming plate. Immediately after collection, the semen should be placed in a water bath at 37 to 39 degrees centigrade before it is handled for evaluation or before it is used for diluting or for other purposes. Now we come to the second aspect of today's talk, the semen evaluation. Semen evaluation is done in a number of ways: the macroscopic evaluation, microscopic test, biochemical test, and in vitro test. More commonly, semen is evaluated for motility, for the sperm concentration, and sperm morphology. If a semen sample is having good motility, high sperm concentration, and the sperms are having normal morphology, then that semen is considered good. For macroscopic evaluation, the color, volume, consistency of the semen are evaluated, and for microscopic tests, the mass activity, individual motility, sperm concentration, live dead sperms, and sperm morphology are evaluated. Biochemical tests include pH, resistance to cold shock, methylene blue reduction test, oxygen uptake test, Resazurin reduction test, fructolysis index, Milanovs resistance test, and catalase test. Some of the in vitro tests include HOST, cervical mucus penetration, acrosomal integrity, and zona penetration assays. Now, first we talk about the color and consistency of the ejaculate. Bull semen is opaque and cream to white colored. Some bulls may ejaculate semen with a yellowish tinge. Buffalo bull semen is white and creamy. It is never yellow. A brownish color in the semen indicates orchitis, and semen with red color is considered serious. The conversion of ejaculates to yellowish green on keeping indicates Pseudomonas aeruginosa infection. Normal bull semen is opaque and creamy in consistency. Semen from most species is homogeneous in consistency, and the presence of flakes or small clumps usually indicates an inflammatory process in the reproductive tract, and the male should be investigated. Such semen should not be used for insemination, and the male should be examined to diagnose and treat the problem. In species such as primates and rodents, semen generally coagulates after ejaculation. Now, based on the consistency and the color, some people say that we can know something about the sperm density, and we can predict something about the number of spermatozoa shown in this chart. In creamy, there will be 1 to 2 million sperms per mm cube. In thin milky, 0.5 to 0.6 million sperms, and in watery, less than 0.3 million sperms per mm. See, now in this picture you can see the color and consistency would vary depending upon the species. You can see here on the left is a sample from bull semen, which is opaque and creamy in consistency, and this is a dog semen, which is slightly watery type. Now, this semen sample shows hemospermia or blood in the canine ejaculate, which is considered a serious problem. Now, this chart shows the semen volume and other characteristics of different animal species, the semen volume, the sperm concentration, the sperm motility, and gel. Semen from a few of the species like stallion, boar, and the camel contains a gel, and we will talk about it. The gel should be separated to use the semen for insemination or other purposes. The semen volume is highest in the boar, 150 to 250 million, and the semen concentration in the bull semen varies from 800 to 2000 million per ml. The gel in the semen needs to be separated out. Especially, you can see in this picture the gel is being separated out for equine semen, whereas the gel in camel semen is very thick in consistency. And when you first examine a semen sample from a male camel, you will not be able to find the motility of the sperm, or you will not be able to find anything. So the gel need to be liquefied by keeping it at room temperature for a couple of hours, and some people and they suggest the addition of enzymes to lies this gel and visualize the spermatozoa. Now, the first uh, microscopic evaluation is the mass activity. The collective movement of sperms or their wave motion is called as mass activity. A drop of freshly collected semen is placed on a pre-warmed slide and examined under a low power of microscope. The you can see here there is formation of uh, swirls and eddies, and it appears that the semen is moving around very fast, and a scale of +1 to +5 is used to designate the intensity of mass activity. Here you can see the mass activity of the spermatozoa. Semen samples are labeled very good when there is very dark swirls and eddies. Good if the swirls are slower and eddies are also slower. Fair, no swirls, but prominent individual cell motion is visible, and poor when there is little or no individual cell motion. We will see in this video how the uh, mass activity appears when we this is a grade 4 semen with very good uh, eddies, and this is grade 3 which is having slow moving waves and eddies. Then after the evaluation of the mass activity, we evaluate semen for individual progressive motility of the spermatozoa. Individual progressive motility of the spermatozoa can be evaluated by manual motility estimates, which is also known as the wet mount. Uh, for this, a 5 to 7 microliter of the diluted semen has to be diluted for evaluating the individual progressive spermatozoon motility, and is placed on a new warm glass slide, creating a drop approximately 3 to 5 in diameter, which is then covered with a cover slip. The volume of semen used, 5 versus 7 microliter for evaluation, will depend on the size of the cover slip preferred, 18 into 18 or 22 into 22 cover slips. The progressive motility of the individual sperms is recorded, and it is a descriptive evaluation. The Here you can see a drop is placed, then a cover slip is placed, and then it is examined under a microscope. The descriptors used are very good. 80 to 100% of the spermatozoa are progressively motile. Good when 60 to 79% of the spermatozoa are progressively motile. Fair 40 to 49% motile, and poor when less than 40% of the spermatozoa are motile. Let us see in this slide the motility of the spermatozoa. Here you can see the sperms are moving forward, progressively motile, and one can appreciate the movement of the sperms, and now one can estimate from the total field how many of the spermatozoa are motile. These scores are, however, subjective, and sometimes some people may have difference in the opinion as to the motility of the spermatozoa. Then there we can use the track motility estimates. Track motility estimates require more time to perform, and images of the sperm are captured by the camera, but they are objective and repeatable. Additionally, one can save the images for a permanent record. Then more recently, there is availability of the computer assisted motility analysis, CASA or CASMA, what it is known as. A number of systems have been developed that allow motility to be analyzed with the aid of a computer. These systems acquire an image in essentially the same manner as described for the track motility technique, and image processing software detects and tabulates the tracks and immotile cells. Other information is readily obtained, including sperm velocity and motion characteristics. Now we come to another important evaluation of the semen is sperm concentration. Sperm concentration is usually evaluated by counting using a hemocytometer, and prior to counting the sperm, they need to be killed to prevent movement. This is often accomplished by adding semen to a diluent containing a small amount of formaldehyde. Now, this is the hemocytometer, and the semen is diluted 1 is to 100. 0.1 ml of semen is diluted with 9.9 ml of the diluter. The hemocytometer is charged with about 15 microliter of diluted semen by placing the tip of pipette in the V-shaped groove of the hemocytometer. You can see here. A cover slip is placed and then the semen is allowed to settle for 2 to 3 minutes. Now the hemocytometer is placed under a microscope, and under a microscope there are 9 large grids visible, and we select the central grid, which is shown as the counting area. The counting area is visible as grid with squares. The central counting area is 1 millimeter square and has 25 large squares, each large further having 16 smaller squares. This large square is having 25 squares, and then each of the 25 squares have 16 smaller squares. The sperms in all 25 squares are counted. All sperms in 4 corner and 1 central square are counted, and the final number is multiplied by 5. Care should be taken while counting the sperm cells on the lines of two sides to avoid duplicate counting. You can see here how counting is being done. This is one sperm, and then you can see here the sperm head is here. This is counted as 2, 3, 4, and then head is here 5, 6, 7, but this will not be counted again in this one. So this will be counted 9, and then 10 this sperm, 11, 12, 13, and then 14. So the heads of the sperm on the margins of the lines are counted. This way we count the number of spermatozoa. The concentration of sperms per ml is derived as follows: the number of sperms counted into dilution factor divided by volume into 1000. Since the volume of diluted semen in the counting chamber is 0.1 microliter, hence to obtain the number of sperms per ml, this should be multiplied by 10,000. Since semen is diluted 1 is to 100 times, hence the number of sperms in undiluted semen will be derived by multiplying with this number with 100. An easier approach would be that the number of sperm heads counted in a single chamber is multiplied by 10 to the power 6 to give the concentration of sperm cells per ml if the dilution is 1 is to 100. Both chambers of the hemocytometer can be counted, but the number should not differ by more than 10%. Then spectrophotometric evaluation is available. A spectrophotometer measures the amount of light absorbed by a sample, and the more sperm are in the sample, the more light is absorbed. By generating a standard curve of absorbance versus sperm numbers, one can quickly and accurately measure sperm concentration without directly counting them. There are, of course, huge differences among species in sperm concentration and ejaculate volume. There is also considerable variability in these parameters amongst males of a single species. Finally, sperm numbers and ejaculate volume are affected by sexual activity. Ejaculates will have fewer sperm when collected during a sexually active period compared to that collected after a period of sexual rest. These types of commercially available spectrophotometric evaluation equipments are available right now, and most laboratories have these equipments to know the sperm concentration. But many a times they validate the findings by counting the sperm numbers using a hemocytometer also. Now we come to the sperm morphology. The basic morphology of the sperms, the live dead count, number of sperms that are live in the sample and number of sperms that are dead in the samples. For this basically a eosin nigrosin stain is used. On a clean glass slide, a sperm drop is made and then a drop of nigrosin stain is placed as shown here. Then one has to mix the semen with the stain, and then we prepare a slide by sliding using another slide. Then we fix it for some time, allow it to dry, and examine it under a microscope. Since live cells, they do not allow the stain to permeate into the sperm head, so they remain colorless. So as shown here, this is a live sperm cell, and this is a dead sperm because it is the stain has passed into the head of the sperm, and which is appearing as pink colored here. So this is alive, this is a dead. In all, 100 to 200 cells are counted, and the proportion of percentage of live sperms is calculated. Then sperm morphology. An important part of any breeding soundness examination is an evaluation of sperm morphology. In the most fundamental case, the size and shape of the head, middle piece, and tail are examined. Additional information can be gained by evaluating integrity of the acrosome and sperm membranes. Sperm from different species vary in size and shape. Bull and human sperm, for example, have paddle-shaped heads. Rodent sperm have hook-shaped heads, and the heads of chicken sperm are spindle-shaped and almost difficult to distinguish from the middle piece. Sperm morphology is usually examined with an eosin-negrosin staining or toluidine staining and examined under a microscope. The abnormalities are classified as primary and secondary. Primary abnormalities are those which arise in the testes, whereas secondary abnormalities arise in the epididymis or ejaculate. The secondary abnormalities may be just as serious as primary. Primary abnormalities decrease through transit and secondary abnormalities increase through transit. Some authors classify the sperm morphological abnormalities in major and minor, which may be considered a better classification. Major include cause early embryonic deaths or prevent fertilization. For example, an acrosome problem prevents zona entry, and a nuclear problems lead to non-fertilization or early embryonic death. Minor problems such as tail abnormalities, example, stop some uh, they stop the sperm movement, so the sperm cannot get to the egg. Then another classification based on the anatomic site of defect. The problem can involve the head, midpiece, or the tail of the sperm. Another approach is to visualize sperm under differential interference contrast microscopy. The sperm are first fixed with glutaraldehyde and can be stored for prolonged periods in that solution. This procedure is particularly useful for assessing acrosomal integrity. Here are shown some of the primary and secondary defects of the spermatozoa. These are the normal spermatozoa. The defects in the acrosome, the defects in the head shape, smaller head, mega head, spindle shaped head, the defects of the mid piece, also the primary defect, and then the double heads or detached heads. They are detached heads are considered a secondary abnormality, and then coiled tails are also considered the primary abnormalities. Then the presence of the proximal droplet as shown here or the distal droplets are considered the secondary abnormalities. Then the detached heads are considered the secondary morphological abnormalities. Primary defects are the more severe and are thought to originate while the sperm was within the seminiferous epithelium of the testes. Secondary defects are less serious and thought to arise during passage through the epididymis or by mishandling after ejaculation. The percentage of sperm morphological abnormalities should not exceed 20% for the semen to be used for further processing. Here you can see different sperm morphological abnormalities, misshapen heads with four normal sperms. This one has a abnormal head, toluidine blue, and large head, elongated head, then the proximal droplet, the distal droplet, the detached head. The bent tail or midpiece in this sperm, and then the coiled tail, the coiled midpiece here, tail in one sperm, and proximal droplet in another sperm. Now, this is the semen analysis nomenclature. Once you have analyzed the semen, what nomenclature it will be given. When the semen volume is none, it is termed as aspermia. When the semen volume is reduced, hypospermia. When the semen volume is increased, hyperspermia. If the sperm concentration is normal, it is termed normozoospermia. If the sperm concentration is reduced, it is known as oligozoospermia or oligospermia. If the sperm concentration is increased, it is known as polyzoospermia. And if the sperm concentration is 0, then it is known as azoospermia, means there is no sperm at all. Then if the sperm motility is decreased, it is known as asthenozoospermia or asthenospermia. If the sperm viability, if there are all dead sperms, no viable sperm, then it is known as necrozoospermia. If the if there is higher presence of morphological abnormal sperms, it is known as teratozoospermia. If the sperm shape only round-headed sperms are visible, then it is known as globospermia. And if the semen is abnormal, there is blood or pus in the semen, it is termed hemospermia or pyospermia. Then we talk about the biochemical tests on semen. Biochemical tests are not routinely performed at most laboratories, but they may be used as a supplementary test uh, for semen evaluation. The pH of the bull semen varies from 6.4 to 7.5, and this can be used as a biochemical test. Then there is a test, the resistance to cold shock. For this, 0.25 to 0.5 ml of undiluted freshly collected semen is taken in a test tube, and it is immersed in crushed ice for 10 minutes. Then the semen is removed and thawed at 30 degrees centigrade, and we perform the live dead count. Better live sperms reflect better resistance to cold shock. Then there is methylene blue reduction test. In this test, the undiluted semen 0.2 ml is diluted with 0.8 ml of agic citrate, placed in a 10 ml test tube, and then 0.1 ml of methylene blue is added. The tube is sealed with liquid paraffin and placed in a water bath at 46.5 degrees centigrade. The blue color, you can see here, blue color is slowly disappearing in 3 to 5 minutes in good semen, and in 9 minutes in average semen, and above 9 minutes in poor semen. Then there is a Resazurin reduction test. For this, 0.2 ml of undiluted freshly collected semen is taken and 0.1 ml of Resazurin solution is added. The solution is covered with liquid paraffin, it is incubated at 45 degrees centigrade, and the color changes from blue to purple to pink and then colorless. Then there is the Milanovs Resistance test. For this, 0.2 ml of undiluted semen is taken in a 200 ml flask, and 10 ml of 1% sodium chloride is added every 10 minutes. After every 10 minutes, we examine the semen for motility and till the motility of the sperm ceases completely. The R test, the R value is calculated by ml of sodium chloride required to cease the sperm motility divided by 0.02. Then there is fructolysis index. Fructolysis milligram utilized by 10 to the power 9 sperms in 1 hour at 37 degrees centigrade. Catalase test and oxygen uptake test. These tests are not used routinely. Then there are in vitro tests to predict semen fertility. The cervical mucus penetration assay. For this, cervical mucus is taken in a test in a empty semen straw, and then this empty semen straw is touched to a semen. The spermatozoa, they travel in the cervical mucus, and the level of travel of the spermatozoa would predict their fertility at to what level they have traveled in the cervical mucus. Then HOST is a sperm vitality test based on the principle that live spermatozoa swell due to the absorption of a hypoosmotic solution. The hypoosmotic swelling test is widely used as a valuable test for determining sperm quality by evaluating the membrane integrity of the spermatozoa of various domestic animals including cattle, horses and swine. The host has also been used as an indicator of the fertilizing capacity of spermatozoa. In this test, 0.5 ml of HOS is taken and 0.050 ml of semen are mixed carefully and incubated at 60 to 120 minutes at 37 degrees centigrade. A total of 100 spermatozoa are examined afterwards under the microscope at magnification of 100 to 400x. The spermatozoa swells And here you can see this is a HOS positive. Red arrows show the HOS positive sperms, and then the yellow arrows are showing HOS negative sperms which have not swollen. Commercially available uh, solutions are available for testing for HOS test for human spermatozoa, and if very few reports uh, indicate their use in animal models also. Then there is the zona penetration assay. It is an in vitro penetration assay. Uh, Another highly successful test was the hemizona assay, where uh, zona is bisected and half of the zona is used for testing the penetration of the sperm through this zona. The hamster ovum penetration test, for this, hamster eggs are used to hamsters are induced to superovulation by injection of hormones, and then the ova are collected and denuded by removing the cumulus and zona. Then they are pre-incubated, and then the sperm is added with that the hamster ovum, and many spermatozoa from many species can penetrate the hamster ovum. The diagnostic value of this test is still controversial, probably owing to the difficulty in optimizing the test protocol, which can lead to false negative results. Then the acrosomal integrity test. The proteolytic enzymes in acrosome digest through zona pellucida, allowing for sperm oolemia fusion. Illustrating this, round-headed sperm indicative of acrosomal absence will not bind to penetrate through the zona pellucida. Staining with different fluorescent lectins that bind to the outer membrane of the acrosomal contents can assess the acrosomal integrity. Now, in recent times, there have been many other assays to predict the potential fertility of the bull semen and the semen from other species. And these tests include the sperm chromatin structure assay, anti-sperm antibody assay, and the evaluation of molecular markers to predict the sperm fertility. Molecular markers, in particularly ubiquitin specific peptidase, Sparta1, calmagene, osteopontin, have been used to predict the fertility as a fertility marker for many species. And this picture shows the work of one of our student who has worked on equine semen and evaluated the Sparta1 and other gene frequencies to predict the fertility. Friends, in spite of a lot of tests available to predict potential fertility of the bull, spermatozoa and spermatozoa from other species, but we have the routinely used tests include the sperm motility, sperm concentration, and sperm morphological abnormalities. You can contact me. This is my email, gnpobs@gmail.com. This is my LinkedIn channel link, and this is my YouTube channel link. Uh, Kindly listen to the lecture completely, give your comments, suggestions, and share and subscribe to the video. Subscribe to my YouTube channel, Govind Narayan Purohit, if you like it. Thank you so much.